So now that we've gotten through the origins of life, let's start getting into the different groups of living things. For this video, we're going to focus on the teeny tiny, the microbes. Now, if you remember, there's some major differences between prokaryotic and eukaryotic cells, and we went into the endosymbiosis theory. There are videos where we talked about that before. I would look at those ones when we look at the cell. But we're going to focus on prokaryotic cells for this video. For the most part, we're looking at bacteria. So bacteria are extremely diverse, but they're kind of a mismatch, right? Some of them are disease-causing, some of them do absolutely nothing for us, and others are actually essential for us to survive. So, for example, some of them help us with digesting our food. Some of them have good relationships with plants so that plants can get better nutrients. And so the term we use to describe bacteria and all the other teeny tiny things is microbe. So we're going to be talking about microbes in general. When you hear the term microbe, it's basically anything that's super teeny tiny that the human eye cannot see. So something you would need a microscope for. And we find microbes in every domain of life, bacteria, archaea, and eukarya, because we're just talking about something you can't see with just your eyes. There is more than 500,000 microbes that have been identified and exist pretty much everywhere on the planet, including some of the most harsh and extreme environments on the globe. We're talking about extreme heat, extreme cold, extreme saltiness. Microbes are very diverse and very hardy. And even when we look at our own bodies, about 90% of the cells on and in our bodies are bacterial. So we can't really fully ever get rid of those germs. Now, bacteria in particular are prokaryotic organisms, so we're looking at something that is simple, small, and does not have a true nucleus. And we identify them based off of their size, shape, how they stain, the colony morphology, and the biochemical reactions they can handle. Bacteria generally reproduce asexually, and they use the process of binary fission to do so, but in order to get a little bit more diversity in the mix, they can transfer DNA in the form of plasmids to other individuals, so they can technically swap small pieces of DNA. Bacteria are very diverse in their abilities to metabolize different types of molecules, and we're going to talk more about oxygen, the oxygen revolution, in the next lecture when we get into things like plants. But there's a lot of bacteria out there that we need as humans. Generally speaking, if it's something that's good for us, we call them probiotics. Anything that is considered pathogenic is disease-causing. Bacteria that are pathogenic can kill millions of people annually, but some of them are always pathogenic, meaning they're always going to cause illness in humans. Others are opportunistic. So Naegleria fowleri, which is a microbe found in warm water, is an opportunistic type of pathogen. It doesn't really ever do anything to anyone unless you happen to inhale this warm water from a warm lake, in which case then the microbe can get to your brain and kind of eat away at it. So in terms of pathogenic versus opportunistic, that's what we're looking at. Pathogenic is it's actively trying to get into your body, trying to cause you harm for its own gain, whereas opportunistic generally won't do anything to you unless the conditions just happen to be perfect. The study of how diseases spread is called epidemiology. And so epidemiology is probably a term you've heard more lately because of COVID, but epidemiology is looking at how do we as humans kind of spread disease around. So epidemiology kind of looks at a lot of different things at once. You're looking at what is the disease, what is the microbe capable of, so what kind of illnesses, what kind of symptoms do they cause, how contagious is it, things of that nature. As well as looking at some of the behaviors that people engage in and how that can change how diseases spread as well. So one of the types of infections out there that we have to keep an eye on and how it spreads are sexually transmitted infections. Now, some of us kind of get a little bit grossed out or feel a little bit uneasy talking about things like that. However, it's important to do that. There's no shame in whether or not you've ever had an STI before, because these are actually really common, and a lot of times you're asymptomatic if you have that. And so when someone finds out that they have an STI, it's important for that person to contact their sexual partners, because their partners might also be infected. Since it's asymptomatic, they might not even know it. So as soon as, if you do find out you have an STI, it really is important to have that conversation with anyone you've had sexual contact with, so that they can get treated to prevent them from infecting anyone else. Basically, in short, use protection, get tested regularly, especially if you have more than one partner or you've changed partners. There's lots of information out there that I'm more than willing to share with you guys to kind of let you know what you need to know about STIs. Now, one of the major issues we're seeing in our lifetime is a good example of evolution in action, and that's bacteria becoming resistant to antibiotic drugs. And it's happening much faster than you would like. So antibiotics are prescribed in specific doses and a specific number of days for a very important reason. The specific dose and length of time you're supposed to take it is given in that format because that's how much it's going to take to kill all of that bacteria. If you don't follow the instructions, if you take less than you're supposed to, if you don't take them for all the days you're supposed to, you run the risk of leaving some of those bacteria behind in your body. So it's extremely important that you complete the full course of antibiotics, even if you feel better. Even if you feel great, keep taking them until you run out. The reason being is if you don't, you're leaving behind the bacteria who are able to survive the first few days of your antibiotic. And if you do that, you're basically increasing the chance that you're developing bacteria that are becoming antibiotic resistant. You'll be looking at this for one of your discussions for this semester, looking at how antibiotic resistance is developing and what that really means for humans. Now, bacteria are one group of prokaryotes, but there's another group called archaea. So these guys are prokaryotes, but they're really different from bacteria. That's why they get their whole separate domain. They have very different DNA, very different chemical composition in their membranes, their cell walls, their flagella. So the only thing they really have in common is that they're prokaryotic and small. But other than that, they're really, really different types of organisms. And archaea are the extremophiles, meaning they like living life to the extreme, meaning in the hottest of hot conditions, the coldest of cold, highly acidic, highly basic or alkaline areas, the really, really salty, the really, really dry areas. These guys are survivors. Now, we can utilize this to our advantage. Some of them are being worked on to be used as bioremediation to help clean up oil spills because they can potentially metabolize the lipids. And then the last group are the protists. The protists is really like a junk drawer of some sort, in that it's a generic term we use to describe all of the eukaryotic microorganisms. So all of the complex cells, like that have a nucleus and organelles, but are still teeny tiny, are the protists. And they were the first eukaryotic organisms. We've actually seen these guys fossilized as old as 1.9 billion years ago. But like I said, protists are kind of a junk drawer. It's a catch-all for all of the micro eukaryotic cells. So they can be plant-like, they can be animal-like, they can be fungus-like. The animal-like protists are going to eat other organisms. The fungus-like ones use spores and they also eat other organisms. And then the plant-like ones, like algae and seaweeds, these guys are going to use photosynthesis. Diatoms are a good example. These are microscope glass-like creatures, because they have silica, that make up a huge portion of plankton, and they're responsible for most of the oxygen in the atmosphere. So these guys actually did kind of do some of the starting work in transforming the atmosphere that we have today. Now, some of these guys can be parasites, and what a parasite is just an organism that's going to live in or on another organism, and that other organism is called a host. So parasites live off of the host, and they basically take advantage of the host. They're going to take things like nutrients. They're going to cause harm to their host. A good example is the genus Plasmodium. These are protists that cause malaria, like Plasmodium vivax, that gets passed on to humans via mosquitoes. So Plasmodium, basically they have a life cycle that is partially in mosquitoes and partially in humans. But now we get to a category that maybe you've thought of, especially in today's world, but didn't really understand what they were, and that is the viruses. Viruses are not considered alive. They're not living things. They're really, really simple in that they're just basically a protein shell and genetic material for the most part. So that protein shell is what we call a capsid, and then on the inside they have genetic material. But even that genetic material could be DNA or it could be RNA, depending on the type of virus. Viruses require that a host do basically everything for them. They require the host for their metabolism, their reproduction, their regulation, and evolution. So this is one reason why viruses would not be considered alive, because they are not a cell, and they cannot do any of those functions like reproduction on their own. They have to basically hijack a cell and make the cell do it for them. Viruses also can be the deadliest of all the microbes. Epidemics have literally killed millions at a time in the past. But they can vary wildly. Some of them are just unpleasant, but they don't cause death, like your common cold. You're sick for a little while, but you get better. Or things like herpes, where you have it for the rest of your life, but it's not going to cause any real harm to you in terms of death or disease. You're just going to be uncomfortable if you have an outbreak. But it can be hard to vaccinate for viruses because they tend to mutate quickly. Consider the fact that you have to get a flu vaccine every single year. So with the flu vaccine, the reason why you have to get it every year is that the flu mutates really, really fast. And you can have many different strains or versions of the flu out during any given season. So the flu vaccine has to change, basically keeping up with the changes the flu has experienced and trying to predict which strains are going to be the most problematic for that flu season. This is one of the reasons why it's so really important to get vaccinated against something like the flu, because it can cause, especially those who are very young, very old, or immunocompromised, can cause some serious illness in people. It's also why we're fighting so hard to get the vaccine for COVID-19. If you've noticed, it's taken quite some time to get a vaccine together, and we're still not done. We're still waiting. That was in March of 2020 when things started to shut down due to the virus. And at this point of recording, we are now in November of 2020 with news just creeping out that some vaccines are seeking emergency approval through the FDA. That is 8 months. And to be honest, that is record time. That is breakneck speed to develop a vaccine that fast. And yet it still feels, for the most part, really, really slow. And we're still waiting because it's not technically done yet. One of the things that we will have to be concerned about is making sure that we keep up the vaccines and keep studying COVID-19 to make sure that it doesn't mutate so that the vaccines stay working. And speaking of COVID-19, sometimes viruses can jump species. That's actually what happened with the coronavirus. So coronavirus is, well, technically a group of viruses, but the one we're looking at is SARS-CoV-2, if I remember the name right. This was originally found in bats and then made the jump from bats to humans. So this is something that's actually of great concern for World Health organizations. Because viruses can jump from one species to another, and because when they jump, we don't necessarily know how bad it could be in terms of, is it going to be really lethal, or is it going to be really contagious? We don't necessarily know that right away. We're really concerned with limiting how many viruses make that jump. And in order to do that, we have to make sure that we are not encroaching on wild places too much. That's one of the reasons why we've seen some of the viruses we see today. It's probably one of the reasons why we have things like HIV. HIV made the jump from apes, from chimpanzees, and whatnot to humans multiple times. And it's because we're coming into contact with those groups, with chimpanzees, or coming into contact with bats. And by keeping us from having that sort of direct contact, we're protecting ourselves from potential new viruses.